There's a dream I feel so rare, so real, and the world in union, the world as one, gathering together, one world, one health, every creed, every color, once joined, never apart, searching for the best in me. I will fight what I can beat. If I win, lose, or draw, there's a winner in us all. It's the world in union, the world as one, as we climb to reach our destiny. A new age has begun. We must face high mountains, must cross rough seas. We must take our place in history and live with dignity. It's our world in union, all divine one, as we climb to reach our destiny. A new age has begun.
Good afternoon, dear our guests. Welcome to our webinar, Ceva Poultry, Demystifying H9N2. So we would like to welcome you here. My name is Dr. Mustafa. I am the Global Marketing Manager in Ceva Poultry, based in France in the headquarters. We are very much excited and happy to have all of you here. Thank you very much for your precious time. Today, we believe we will be talking about a very important subject as Ceva, as science and innovation-driven company, and we will be talking about the most popular topics nowadays about H9N2. And I will be your moderator today. So when I look at the numbers, we are really increasing, and thank you very much. It's very important for us to see all of you here. And I believe we will be really meeting your expectations about this meeting. Today we will have 5 speakers and 3 presentations, and I would like to give you a bit guidelines about our webinar today. So you can see on your right screen a vertical bar, and you can see the Q&A tab. So I kindly ask you to ask your questions at any time while clicking that Q&A. And if you see any questions has been asked already by somebody else, you can feel free to like this button, so we will be able to see the relevance of this question as well. So about the answers, we would like to answer all the questions at the end of the sessions. So I kindly ask you also to mention the person you address this question as well. So thank you very much again, and I would like to today invite our first speaker, Dr. Anand Vatkar, the Country Director, India and South Asia. Dr. Anand?
Thank you, Dr. Mustafa, and good afternoon and welcome to this webinar, all the participants. We thank you for your time. It's an apt time for this webinar, which is named Demystifying H9N2. My name is Dr. Anand Vatkar, and I am the Country Director for India and South Asia for Ceva. As you know, Ceva is one of the fastest growing top 5 animal health companies based in 46 countries and working across 110 total countries. Of the 4 values that we are guided by, 2 of them are directly linked to today's webinar: customer passion and innovation mindset. If you have used Ceva's products or services anytime in the past, you would have experienced our customer passion. And that brings me to innovation mindset. We use innovation mindset to solve customers' problems, to create long-term value through the solutions which we create jointly with our customers. And this webinar is a shining example of such an co-created solution, that we want to share the knowledge that we have gained across different parts of the world and what we know about this disease that affects our country. With that, I will hand over to Milind Limaye, our technical director based in Pune. Thank you.
Good afternoon and welcome, dear guest. It is really very exciting moment for me to see a galaxy of veterinarians and decision makers attending this presentation. It indicates that we are very serious. This is just in keeping with the legacy of Ceva, where we mainly disseminate knowledge, the knowledge which will enrich our industry. I was very excited also because Honorable Animal Husbandry Commissioner of India, Dr. Praveen Malik had agreed to join, and he was looking forward to have a keynote for us. But unfortunately, he's not keeping well today. I think he's running a little bit of fever and due to overstress probably. So he has apologized. He could not attend this, and we are missing his keynote. I'm sure in the time to come, all of us will get a chance to listen to his understanding and his insight about the problem that our country is facing right now. He has got a very clear and scientific view. I really appreciated that. And he was quite keen to join this webinar. However, unfortunately, he could not join. Nevertheless, I can assure you that this is going to be very enriching presentations for you. Without much further ado, I would like to give it back to the moderator. Thank you very much.
Thank you very much, Dr. Milind. It's very much important. So now it's time to talk and invite the first speaker. I would like to invite Dr. Güvenel Dauphin. Dr. Güvenel, you can share your screen now.
Good afternoon, everybody. I'm very happy to be able to give this presentation to you today about the worldwide perspective on this H9N2 virus and disease. So I would first like to start with some key points about H9N2 context. First, this H9N2 subtype was first detected in 1966 in turkeys in the US. It has been well established only the last 20 years, in domestic chickens. Third point is that this is always a low pathogenic influenza. And fourth point, it has more economical impact than highly pathogenic avian influenza, at least on the Indian subcontinent, although it has attracted less attention in disease management and public health control as compared to H5, HPI viruses. But this is actually quite changing the last years. And you can see it from the publications that we can see in the public domain, including these 3 major publications, review articles published by quite famous avian influenza scientists who look at the zoonotic aspect of H9N2 as well as the fact that it is an emerging challenge and continuing to grow in the poultry industry. So let me start with the global H9N2 situation. Here is an FAO map from their database where you can see the reports of avian influenza outbreaks officially reported to the OIE as well as on national websites. And if you look at the legend on your left-hand side and the color-coded dots, you can see that you have a vast diversity of different subtypes. And if you look at the H9N2 dots, which are located here, actually, you can see that you only have a few H9N2 cases among all the avian influenza outbreaks. And these were some H9N2 human cases located in China within that period of 2 years. So if you look at an FAO, again, an FAO map from their EmpressI database, only looking at H9N2 reported outbreaks in a longer period, which is more than 10 years, you can see that the number of dots is actually not very high, and higher numbers in Asia. So the prevalence of H9N2 virus is not very clearly known. And the reason is that this is not a notifiable disease. So countries just don't need to report to the OIE. And even at the national level, in most countries, this virus doesn't need to be reported to veterinary authorities when detected. However, we think it is the most prevalent and widespread influenza subtype circulating in chickens, and it is endemic in poultry across Asia, across Middle East and Northern Africa. So this is a map extracted from a publication issued in 2017, and at that time you could already see quite a high number of infected countries, and the whole story in Asia started in China, and then the virus progressively was spread down south and west, and it progressively reached the South Asia, the Middle East, and now Northern Africa. And since the publication was actually issued, we had Algeria becoming infected, several countries in West Africa, and also 2 countries in East Africa, Kenya and Uganda. And also further south, Malaysia also became heavily infected. This is quite a nice study that actually describes the genesis of avian influenza H9N2 viruses in Bangladesh. And these researchers actually looked at virus sequences, and they tried to trace the history of the virus circulation, combining virus sequences and virus evolution over time and across geographical areas. And the hypothesized roots that they found was that some H9N2 virus left from Hong Kong in 1997, and it flew to United Arab Emirates as well as to Pakistan. These were this was the same genotype, probably because of poultry trade. After that, after 1997, the virus from Pakistan also flew to Japan because of pet trade. And then they also identified some virus from Iran, which went to Pakistan in the years early 2000. And then in Pakistan in 2004, they noticed that looking at sequences again, that some viruses, H9N2 viruses, reassorted with the circulating H7N3 virus at that time. And this gave birth to a set of different viruses, of chimera between those viruses. And some of these viruses went into India and later on to Bangladesh. And then some H9N2 viruses became established in Bangladesh. If we look at a similar study, which that time focused on Egypt, but also looked at a spread across Asia, Middle East, and Northern Africa, they actually looked at sequences from 700 viruses over a period of about 10 years. And we found that the Middle East region actually acted as a platform for H9N2 viruses, because they could see that some viruses went from Middle East to Egypt and Israel and came back to the Middle East, and other viruses also were spread to Iran and India and China. So they hypothesized two possible causes for this. First, of course, poultry trade among these countries, and also the fact that this region is also on the way for wild birds migrations. Now, looking at the Indian context, this H9N2 virus was first isolated in India in 2003-2004. If you compare to China, it is far after China, where it was reported for the first time in 1982. Then, if you look at the scientific literature which was published on the Indian situation, I only found 13 scientific papers from international peer-reviewed papers. And if you compare to China, which has over 500 papers, this is not a lot of scientific literature. And if you look at the virus sequences that have been deposited into the public domain, there are only 48 Indian isolate sequences, and among these, one from the past 5 years. And China has over 9,000 isolates, half of which have been deposited within the last 5 years. And so we at Ceva, we looked at some sequences from these three countries and built this phylogenetic tree. And what you can see first is that you have clear different clusters between Bangladesh, India, and Pakistan. And if you look at the Indian strains, this group actually clusters with this Pakistani group. So it shows that there are clear cross-border exchanges of sequences among countries. And if you look at this sequence in India, it also clusters with Bangladeshi sequences. And you also have a cluster in the middle, which seems to be dedicated to India only, and further away from strains from in Bangladesh and Pakistan. So it would actually be very nice for India to sequence more H9N2 viruses, and this will enable to understand better how these viruses circulate within India and also with the neighboring countries. So now looking at virus diversity, first I will quickly describe how this virus evolves. So this is a virus particle, an influenza virus particle. It has two surface proteins, namely the hemagglutinin and the neuraminidase, and these proteins are the basis of the subtype labeling. You can have 18 different HA subtypes and 11 different N types. And in addition to these surface proteins, you have 6 internal proteins. Some of them help maintain the structure of the virion, and others help the virus to replicate within the cell. And so this virus also has 8 segmented RNA, and these RNA segments actually live independently from each other, which means that they can evolve separately, independently. And these RNA viruses have 2 very efficient modes of evolution. The first one is the evolution based on the replication of these RNA segments. Because it is an RNA virus, the RNA polymerase induces a lot of errors, and these errors are opportunities for the virus to mutate. So this is a phylogenetic tree of different sequences of HA gene, and you can see from this tree the evolution of viruses. So the longer the branch, the higher rate of mutation, and you can see here very nice and regular mutation of these viruses. So this is what we call the drift, genetic or antigenic drift. These are minor mutations, but it allows the virus to slowly mutate and adapt to new situation. There is a second mechanism of virus evolution, which is a more drastic one. It is called reassortment. When you have two viruses infecting the same host cell, these viruses, when they have replicated and they are ready to form a new virus particle, the virus particle which will be formed can be made of any of the segments originating from these two different viruses. So you will end up with a chimera of RNA segments. And this is a very efficient way for the virus to evolve drastically. And these reassortments can happen within a given subtype, for example, within H9N2 genotypes, and it can also happen between different subtypes. And we know from the last 5 years or 10 years that H9N2 is a very active virus in terms of giving genes to other subtypes, and it has actually helped other subtypes to emerge and to even become zoonotic. That's what we see from this Chinese publication, and we know it has been a gene donor, a segment donor from its internal gene to all these viruses, including the H7N9 virus, which emerged in China in 2013, and also including this very famous HPI-H5 virus that we all know about. So coming back to this publication on the genesis of H9N2 viruses in Bangladesh, this is how they actually reconstituted the scenario, this scenario of spread of the virus. They actually looked at the different segments, the sequences in these viruses. They looked at this over time and also correlated it to the geographical location. And they actually came to this scenario. So you can see here what I mentioned about the reassortment of H7N3 virus with H9N2, which generated many different possible viruses, but only one of them jumped into India at that time between 2004 and 2009, and then spread further into India and Bangladesh. And other similar events happened also the following years. And this is a constant dynamic within countries. And this other paper, which focused on Egypt, actually rather looked at the HA gene and its evolution, and they made the correlation between the virus, the gene evolution, and the geographical location. And you can clearly see from these two figures that you have virus clusters, phylogenetic groups that are correlated with the geographical location. So Northern Africa and Middle East here would have their dedicated viruses. So a virus would be introduced into a new region, and then it would evolve locally. If you look at this overall virus evolution, so this is an extract from a very nice application which is called Next Strain, and this is an alignment and phylogenetic tree of all H9N2 viruses across the world. And H9N2 viruses actually have two main lineages. One is the American lineage, which is here down with North and South American lineage, and then all the other viruses are Eurasian lineage. And this very large lineage has sub-lineages, and you can see also, as was also stated on the last slide, that you have a very clear correlation between a sub-lineage and the geographical location. And so these are the sub-lineages that are well known. There's the G1, which is now divided into Western and Eastern, mostly circulating in Middle East, Egypt, and also Bangladesh and India. And then we also have a Beijing 94, also called Y280, which mostly circulates in China and Vietnam. And we also have Korean and the Y439 in Eurasia and Korea. So this map tells you gives you the same information than before, but a bit more clearly. What you can see from this map is that the G1, Western G1, is the most spread geographically, is the lineage which is mostly spread. And another comment that I would have is that the human cases from H9N2 are always from the Beijing 94 sublineage. So now, how about the international regulation and coordination on H9N2? OIE has a chapter in the OIE code on infections with avian influenza viruses, and you can hardly read about H9N2 viruses in this chapter, and I'm going to explain why. It is because of the classification of avian influenza viruses. So if you face some outbreaks with poultry or white birds, and if this outbreak shows to be a highly pathogenic, that means at least 75% mortality, or if you face an outbreak which does not where high mortality does not occur, but we know that it is linked to H7 and H5, then you have a risk for reversion to virulence. Then you have to notify this outbreak to the OIE. On the opposite, if you have an outbreak linked to low pathogenic avian influenza, and this outbreak is not related to H5 or H7, then it is not notifiable to the OIE, and this is the case of low pathogenic H9N2. And this classification has great implications in terms of disease surveillance, prevention, and control, because in the first case, the veterinary authorities will have to get reports, and they will have to intervene. They will have to go for stamping out, compensation, and they will have to monitor the disease situation. And if they embark on a vaccination, they will also have to monitor the vaccination and run a strategy for vaccination. While with H9N2, of course, veterinary authorities will have to decide to authorize vaccination or not, but usually surveillance, prevention, and control are done by the poultry industry itself, usually collaborating with universities. So this H9N2 virus is also now under the scrutiny of the World Health Organization because of the recognized zoonotic risk of this virus. And twice a year, antigenic influenza data as well as genetic data are gathered for human seasonal influenza as well as zoonotic influenza. And for zoonotic influenza, the OIE-FAO network of expertise on influenza would also help WHO to gather this data. And this is actually the list of candidate vaccine viruses for H9N2. So these viruses, these strains are kept by the WHO and its collaborating centers, and these strains are just ready to go for vaccine production in case there was the beginning of a severe human epidemic or pandemic. In addition, WHO runs quarterly risk assessments at the human-animal interface for influenza, and from the last years, they started including H9N2 in their risk assessment bulletin as well. So here are my conclusions of this presentation. First, H9N2 is a major economic threat to poultry industry, and it also has zoonotic implications. Second, H9N2 viruses continue to expand to more and more countries, and they continue to diversify into multiple lineages, sublineages, and genotypes. And it is a very active circulation. Third, the spread of these viruses between and within countries is part of a complex migration network from far Asia to the Middle East and to Africa. Monitoring and characterizing H9N2 viruses will help understanding how these viruses spread, how the migration patterns look like, and this will help and provide useful guidelines for virus and disease control. And finally, the international scientific community states that the control of these viruses in endemic country is necessary to reduce both the spread in animals and also in humans. Hence, it is the responsibility of veterinarians to protect both animals and humans. With this, I'm going to thank you all for your attention, and I'm going to be ready for your questions at the end of this session. Thank you, everyone.
Thank you very much, Dr. Guvenel. It's very much clear, and now you gave us a lot of ideas about the worldwide perspective of H9N2, and I believe it's very much relevant after hearing your presentation why we are organizing this webinar. So now, before I invite Dr. Christophe Casaban, I would like to remind all our valuable attendees to keep asking their questions on the Q&A, but as I mentioned before, we will be answering all of them at the end in order to avoid any interruption. So now the floor is for our valuable expert, Dr. Christophe Casaban. Dr. Casaban is 52 years old, married, and has 3 sons. He has been graduated from French University, Veterinary University of Lyon in 1993. He completed his education by specific training on virology, immunology, and avian pathology in Lyon and Mayon Alford universities. He started his professional career in poultry biologicals right from the beginning, 1994, in Rhône-Méry, which later became Meriel, and now it's Boehringer Ingelheim. In 2002, he joined Ceva Santé Animale as International Technical Manager for Poultry Vaccines. After 4 years in this position, he moved to Poultry Range Manager for an area covering Turkey, the Middle East, and the whole Africa. From early 2020, he has been appointed as Global Scientific Director for Poultry Vaccines. His responsibilities include providing scientific expertise to the field, managing scientific investigation studies, and networking with global key opinion leaders. He issued several papers about Gumboro disease prevention, vaccination techniques, pig quality, Bursa Fabricius physiology in international scientific journals or congresses. Now, the floor is yours, Dr. Christophe Casaban.
Thank you. Thank you, Dr. Mostafa. Warm welcome to all of you, dear guests. I send you my greetings from France. My name is Christophe Casaban. I'm the Global Poultry Scientific Director for Ceva Animal Health. So after Dr. Guvenel helped us to understand a little bit the overall epidemiological situation of H9N2 in India as well as beyond in the rest of the world, as well as the type of regulation that is ruling this type of infection. Let's get a little bit closer to the chickens, and my talk will be about stressing why and how this pathogen is indeed quite important in chickens in terms of disease, in terms of disease impact. So I will subdivide my talk into a few chapters just to insist on few important aspects that are important to share with all of you. First of all, we have to understand that by itself, H9N2 can be quite harmful to chickens. Not very, very common. We are most likely facing some co-infections, but it's important to bear in mind that in some cases, H9N2 alone can be responsible for some very strong clinical manifestations. As you can see from this picture, this broiler is displaying a very strong edema of the head, of the different subcutaneous tissues of the head. and we hardly see its eyes anymore. Obviously, this also looks like Mycoplasma gallisepticum or pollen head syndrome or infectious coryza, but let's say it's definitely one of the major sources of upper respiratory tract infection. So we have to put H9N2 in the list of our differential diagnosis when we observe this type of sign in the field. Another quite important aspect of H9N2 harmful effect is the ability of this virus, even alone, to induce a quite strong drop in egg production in breeders as well as in layers, as you can see from this graphic. I had the chance to attend a seminar in Bangladesh that was organized by Ceva and the ACI 2 years ago. And there, the local representative of the Bangladeshi Livestock Research Institute orally mentioned what was the direct economical impact of H9N2 outbreaks in his country. And I took notes of his figures. So we can regard them as official figures. As you can see, H9N2 can dramatically affect the market as well as market prices just because of the drop in the chick production. Remember the drop in egg production. So as a result, you are getting less eggs, and also there is a drop in hatchability. So altogether, it will induce a drop in availability of the chicks. As a result, obviously, with a shortage of the chick supply, now the prices are increasing. You can also notice that there was also a rise in production costs, and again, as I mentioned before, a drop in egg production by 30%, which is obviously quite significant. The other source is coming from Indonesia, and this is a written paper which is providing some other figures, in this case in layers, and you can see the egg production drop as well as mortality. However, in this case, the authors are underlying that in 70% of the cases, H9N2 was alone, but in 30% of the cases, we were observing some mixed infection with other important respiratory pathogens. So in this case, it was not only H9N2 in 100% of the cases, but all in all, a very strong impact on health as well as on economical return on investment for this production, either meat or egg production. There is another very important feature of H9N2 virus is its amazing ability to induce immunosuppression. There I found at least 2 publications, including a very recent one, which are clearly describing how H9N2 virus is able to specifically induce some damage to the thymus as well as to the bursa fabricius. And as you know, these are the 2 primary lymphoid organs of the chickens that are ruling the T cell response and as well as the B cell response, respectively. So H9N2 can induce degeneration of these 2 organs, as well as obviously a lymphocyte necrosis, which means that ultimately the birds will get less white blood cells production. Also a drop in organ indexes, including spleen. By flow cytometry, we can also measure a significant decrease the 2 main population of T lymphocytes, and ultimately, as a result, these authors were able to show a significant reduction of vaccine response. So we still don't understand exactly why H9N2 is able to induce this type of signs, but it's very clear that among the overall huge family of avian influenza viruses, this particular subtype is very critically prone to induce by itself these immunosuppression effects. So this is something very important, I would say, to bear in mind. What is the most widely experienced situation is a combined infection of H9N2 with another respiratory pathogen. And it seems that these two guys are acting synergistically towards getting an even more acute or more severe clinical manifestation. And what's interesting is that some authors, they succeeded to reproduce it in experimental conditions, just to confirm that, yes, indeed, what we observe in the field is actually the reality. In this experiment, these authors, they inoculated either H9N2 virus alone, so this is the dark blue line here, or E. coli suspension alone, that's the green line here, or both together, E. coli and H9N2. This was in broilers, and this is the red line here. So what we are observing is, on one hand, a much higher clinical score in terms of, respiratory manifestations, as well as a much longer time span where these birds are suffering from respiratory infection. So there is definitely a synergistic effect of E. coli together with H9N2. The same applies with infectious bronchitis virus. So in this case, these were authors from Morocco, so they used an isolate from Morocco. And again, by checking the group receiving the IB virus alone, the influenza virus alone, or as highlighted here in this red rectangle, the group receiving both viruses, we could very clearly see an increase in the intensity of respiratory signs, as well as more intense histopathological lesions of the overall trachea and respiratory system, as well as necropsy signs. So again, a synergistic effect of these two viruses for the respiratory system. This can be evidenced, and this is almost diagnostic, I would say, when we observe at the bifurcation of the bronchi these massive plugs of fibrin in these two bronchi. This is quite typical for a combined infection of H9N2 virus together with infectious bronchitis virus. So obviously, these birds will eventually die because they cannot breathe. Simple like that. What is very interesting is that in the recent literature, there have been already some explanation on the why this synergistic effect. And especially when it comes to bronchitis virus with H9N2, it seems that indeed bronchitis virus is able to help H9N2 virus by providing trypsin in order to help the influenza virus to replicate faster. So that's very interesting to see and to currently understand what are the mechanisms behind this combined synergistic effect of these two viruses. And indeed, when I looked at the overall bibliographical references on these potential cofactors, H9N2 can come along with E. coli, with Avibacterium paragallinarum, so the agent of infectious coryza, or Staphylococcus aureus. It can come together with Mycoplasma galisepticum, Chlamydia psittaci, or viruses, especially infectious bronchitis, as well as highly pathogenic avian influenza virus. This is quite well described in Egypt, for instance, as well as Newcastle disease virus. I'd like to draw your attention to the fact that this can also apply to respiratory post-vaccination reactions to strong vaccine virus for Newcastle disease, including Lassota, for instance. So when we are facing birds that are suffering from post-vaccination reactions to Lassota vaccination, and when H9N2 virus comes that time, then we can get a very complicated respiratory syndrome due to this combined infection. Human cases, Dr. Gwenel already mentioned that, so this is just for your records. And just to keep that in mind, for instance, this recent paper in emerging infectious diseases from the CDC of Atlanta, disclosing a case in India where there has been a clear mixture of H9N2 together with some genes from a poultry virus. So just to keep in mind that this virus is also under the radar of the World Health Organization, for instance. Last point, it has been already mentioned by Dr. Gwenaël, but I would like to insist on that. H9N2 virus seems to be extremely generous, if I may say, in such a way that it likes share its pieces of RNA genome with other avian influenza viruses. And by this way, it seems that H9N2 in particular is the one of the responsible for the genetic drift and shift of other influenza viruses. So two well-described examples from the literature. As you can see, if you move from the top to the bottom of this graphic, you will see that the ancestors are well known to be H9N2 together with H5N8, which was a highly pathogenic poultry virus, of course. And then by mixing RNA segments of the genome, because of this combined infection in the chickens, we ended up into a completely new virus, which was H5N2, which was still highly pathogenic, affecting chickens in Egypt. But we can now clearly understand the past history, or what are the ancestors of this virus that emerged in this country in the way of H9N2 and H5N8. But that's why it gives hard time to the local authorities and veterinarians there, because we are able to observe constantly newly emerging viruses in this country due to this frequent mixing of genes. Another very interesting phenomenon was around H7N9. As you probably remember, when this virus emerged in China first, it was low pathogenic in chickens. However, there have been very quickly reports of cases in humans which were quite severe. And we know now that indeed this H7N9 virus contained H7 genes as well as N9 genes coming from waterfowl and poultry, but the other 6 genes were derived from H9N2. And these particular genes helped this H7N9 virus to infect humans. We know from that time that nowadays this H7N9 also switched towards a highly pathogenic avian influenza subtypes for poultry. So you see there has been a kind of double evolution of this virus. But again, definitely its ability to infect humans came from these H9N2 genes. So as a conclusion, the take-home message from my talk on the importance of this H9N2 in chickens, first of all, it can be pathogenic and quite pathogenic in chickens, either directly through the respiratory system or through drops in egg production in breeders and in commercial layers. Definitely, it's very clear that it acts synergistically with other respiratory pathogens. It's not always clear which one came first, if it's H9N2 by itself because of its immunosuppressive capabilities, that is opening the door to secondary infective organisms or microorganisms that will infect in an opportunistic way, or if they combine together, we still don't know, but definitely there is a synergism of H9N2 with other respiratory pathogens whenever it's bacteria, mycoplasma, or viruses. Again, it can be zoonotic by itself. And last point, very important to keep in mind, it's able very strongly to act as a gene donor to other avian influenza virus subtypes. And by this way, I thank you very much for your attention, and I'm looking forward to answering your questions. Thank you very much.
Thank you very much, Dr. Christophe. Thank you very much. It was very clear. So you talk about the importance of H9N2 in 4 main pillars. I believe it was very clear. Again, I kindly remind that you can keep asking your questions on the Q&A on the right hand of the screen. Now I would like to invite our next speaker, Dr. Marcelo Paniago. Dr. Paniago is our Director, Asia Veterinary Services. He's a veterinarian graduated in Brazil and subsequently with master's degrees in preventive veterinary medicine and business administration. He has worked in the university as a lecturer in poultry diseases, and then he moved to Agroques Ros, today Aviagen, in Brazil, where he spent 10 years. He joined Ceva Animal Health in 2004, initially in Asia, then in France, and since January 2017 for the Veterinary Services Group of Ceva Animal Health Asia. He has worked in poultry sector for over 34 years. Wow. Dr. Marcelo, now the floor is yours.
Thank you. Thank you for the introduction. And it's a pleasure to see many of you in this seminar. And I hope that we can have a fruitful discussion after all these presentations. But my target today is really to discuss some strategies to control H9N2. And before starting this presentation, I would like to share something that happened to me about 15 years ago or even longer. In those days, if you might remember, everything was around H5N1. Everybody was really worried about H5N1, the losses caused by that virus back in 2004, 2005. But then I went to Indonesia and I met a very well-known veterinarian there. And he told me that, well, Marcelo, you will see today we are facing this problem with H5N1, but you will see that really the virus that will cause a havoc, the virus that will cause problems will be the H9N2. And honestly, I have to confess that when he told me that, I found it hard to believe, but it's true. And today I have to recognize that he was right. 15 years ago, 16 years ago, when he told me that, he was right. And today we're facing huge problems related to H9N2, right? H5N1 is still present and highly pathogenic avian influenza is still a problem, but H9N2 is really causing serious troubles to producers, right? So this is an issue that really I really like, and I would like to start my presentation by just reminding some of the topics that were mentioned before my presentation here. H9N2 really causes severe losses to the poultry industry. I don't need to revisit these topics, but we all know that this is a serious issue that we have to deal with. Another point that was mentioned before, that H9N2 is known to be zoonotic. This is another factor that makes this disease, this virus, so important to be well controlled. And it was also mentioned that H9N2 is a gene donor to other avian influenza virus, like H5N1, H5N6, H7N9, H10N8. And just to give you some figures about the H7N9, the first time that this virus was reported was back in 2013 in China, in the southern part of China. And today, this virus has caused the death of more than 1,500 people and only circulates in a small area of China. To have a comparison, the H5N1 that is somehow endemic in some countries in Asia since 2003, 2004, this H5N1 has caused approximately a little bit less than 500 deaths. So This H7 and 9 that has some genes come from H9N2 has caused 3 times more deaths, human fatalities, as compared to the H9N1. So it's really a virus that we have to keep one eye open. And finally, it was mentioned in the first presentation, it is widespread. It goes from all the way from Indonesia to Middle East and to northern part of Africa, and now is moving towards the southern part of Africa. So it's really a huge geographical area. But I would like to add one more thing, one more point to address. When we think about eradication of this virus, let me make a parallel with highly pathogenic avian influenza. Several countries have been affected by HPAIV. It might be H5N1, H5N2, H5N6, or H5N8. But several countries have been able to eradicate. Malaysia, the country that I live in now, has been able to eradicate this virus 6 times. The Philippines, Thailand, South Korea, USA, Italy, Netherlands, Germany, England, Poland, France, and you name it. There are dozens of countries that have been able to eradicate this virus. Of course, some reinfections might have happened, but the countries usually are able to eradicate this virus. But now, look at the list of countries that have been able to eradicate H9N2. After this virus becomes widespread in a country, what it becomes entrenched, there is no one single country today that has been able to eradicate this virus. So the question that we have to answer is that if eradication is not feasible with the tools that we have today, what could be done to reduce the losses associated to H9N2 virus? What tools that we have in our hands to reduce the losses, to mitigate the severe economic impact of this virus? So to answer to this question, we need to understand how this virus will cause an impact on the poultry industry. I will show some slides that my colleague Dr. Casaban has shown before, but the virus itself, the virus alone, is able to cause severe impact, and this was a publication on the G1-like lineages, the one that is circulating in India, and it can cause severe drop in egg production. This was in experimental conditions, but the virus itself might be able to cause. It's not the most common ways. Usually it's associated to something else, but even some of them are able to cause that. But when the virus is associated to other pathogens, then the situation becomes even worse. I just brought here one paper published in India, but Dr. Gautaman and his co-workers. And in this article, they mentioned that when there is only Newcastle disease virus, you can see here the mortality is 7.75% in layer flocks. However, when there is a co-infection with LPAIV, low pathogenic avian influenza, plus Newcastle disease virus, this mortality goes to 19.7%, highly significant difference. So you can see that when we have another co-infection, the cases are much more severe. My colleague, Dr. Casaban, also mentioned the association of H9N2 with an excessive vaccine reaction. Yes, this is well published in the scientific literature. And here he mentioned the Lasota strain. And by coincidence, I brought an example of the association of the H9N2 plus Lasota. You see the severity of the clinical signs of the vaccinated birds compared to the birds that were infected with H9N2 alone, or the birds that only received the Lasota vaccine. So you see that there is really a kind of synergism between the H9N2 and an excessive vaccine reaction, in this case called by Lasota. But there is also documented for other vaccines, ILTV, laryngotracheitis vaccines, for example, and others. And finally, H9N2 and environmental factors. Also, there is some descriptions that some environmental conditions can be stressful and can aggravate the infections by H9N2. And two of them are high temperature or high ammonia levels. These two conditions can aggravate the problems related to H9N2. So after said that, when we think about how to prevent or how to reduce the impact of H9N2, we should consider to fight against all those cofactors. So the first one, and probably the most important of them all, is biosecurity. If I have a risk of infection, I have H9N2 here, and I have my farms, it can be a broiler, breeder, or even layers. If I have my farms or my flocks here, the first layer of protection is biosecurity. If I'm able to keep this virus outside of my farm, of course, I will not have losses associated to it. Sometimes, or most of the times, this is not possible due to some conditions in the farms. This is not possible. Then we should do something else, like prevention of immunosuppressive disease. Is also, and Dr. Christophe mentioned that, well defined that immunosuppressive disease can aggravate infection of H9N2. So prevention of immunosuppressive disease like IBV, CIAV, and others. Something else, we should vaccinate against other respiratory disease. If IB can have an impact on the cases caused by H9N2, I should have a good vaccination program against IB and against NT and against laryngotracheitis and so on and so forth. I should consider, I should aim at reducing the post-vaccination reactions. So there are several points to consider. Environmental conditions, improving environmental conditions is one of them, and actually vaccination against H9N2. So at the end of the day, there are several points that I have to consider, several shields, several things that I should do to reduce the impact of H9N2. One thing that we have learned that when we depend only on vaccination, the chances of failure are significant. So in countries where vaccination is allowed, it's recommended to associate vaccination, but with improvement of environmental conditions and the other topics that I mentioned before. Okay, it's not only the vaccination that would solve the problem. But then I have to answer something. Okay, let's discuss about vaccination. Probably is the topic that everybody wants to discuss, and we don't have time to go deeper in the other topics. But in terms of vaccination, well, although let me read this quote from this researcher from South Korea, I'm sorry. And this author, he mentioned that although immunization with inactivated LPAI vaccine cannot completely protect against viral infection and shedding into the environment, vaccination is one of the most promising control measures to date for H9N2 LPAI. So this was mentioned last year in the publication of last year. And there are several countries actually that are officially vaccinating, and you can see the list of them here: China, South Korea, Pakistan, Iran, Lebanon. I'm not going to read, but you can see that several countries today adopted vaccination in an official way. Well, but then the next question is, what can be expected from vaccination, right? It's only protection, clinical protection, but what, from a country perspective, what can be expected from vaccination? Well, I'd like to bring a publication again from last year, and this publication addresses this issue in Korea. Okay, so just for you that are not familiar, some of you that are not familiar with South Korea, it's a small country, but there are almost 1 billion broilers per year. is a huge layer industry, and this country has a consumption of 15 kilos of chicken meat or 10 kilos of eggs. And interesting to mention that this country was able to eradicate highly pathogenic avian influenza 6 times. So it's a country that has experience in eradication, has the money to do compensation, and they were not able to eradicate H9N2. But let me focus in this publication. Well, what this publication says is in a very short summary, is that in 1996, there was the first outbreak of H9N2 in that country. Between 1996 and 1999, the Korean authorities tried to eradicate the virus by stepping out, by doing compensations, but this policy did not achieve complete eradication. So for several years, nothing was done from the official point of view. But then in 2007, the government decided to allow vaccination, and they even have an official vaccine strain that was distributed to 5 laboratories, and they produced the vaccine from this vaccine strain that was given to them by the government. So since then, since 2007, the vaccination has been a routine in that country. In this particular article, they assessed the vaccination from 2007 to 2017, 10 years, 11 years of vaccination, right? So what these guys saw is that first, they had a good vaccine coverage. They sold almost 700 million doses in that period from 2007 to 2017. And it officially covered 50% of the layer chicken farms and 50%, nearly 50% of the breeder chicken farms. So this is what this paper is saying. So it's a reasonably good coverage with this vaccination. But then what was the result of this vaccination? So the first point to understand is that according to the regulation, 20 to 30 unvaccinated sentinel birds, they have to be co-housed with the vaccinated flock for 3 to 4 weeks. And then again, according to the regulations, serum samples from 10% of the vaccinated farms have to be analyzed. And in this 10% of the farms that are analyzed, they would take samples from the birds that were vaccinated, but they would also take serum samples from the sentinel birds. So this is the way they evaluate the progress of the vaccination in that country. And the results were interesting. In 2007, the H9N2 infection occurred in 18% of the vaccinated farms. So it gives around 1 farm out of 5, they still have the virus circulating. In 2008, so 9% of the farms, or in other words, 1 out of 10, 1 out of 11 farms, they still detected the H9N2. And from 2009 onwards, no H9 infections were detected, were reported according to this methodology. So all the samples from sentinel birds were negative. So in fact, there have not been reports of H9N2 outbreaks in broiler farms in Korea recently. There is another publication that reports only from 3 unvaccinated live bird market in 2017 and 1 case in 2018. So according to this publication, the vaccine was able somehow to reduce significantly the circulation of the virus, reduce the loss associated to it, right? Well, but then a question that we should try to address is that, will vaccination against H9N2 increase the virus mutation rate? Right? This is something that is in our mind. We are going to vaccinate birds. This vaccination will increase the virus mutation. Well, let me bring to you three different research to try to measure this mutation rate in the H9N2. The first one was this publication in 2018, and the researchers did something that was interesting. First, they produced a vaccine using one strain called F98. This strain was isolated in 1998 in the southern part of China, in Shanghai to be more precise. Well, and they produced a vaccine from this experimental vaccine from this virus. And then they vaccinated some SPF hens to the point that these SPF hens reached 9 log 2 of titers, right? And after that, they had also SPF hens that were not vaccinated. So they mated these flocks, these birds with males, of course, and they collected the embryonated eggs. So, of course, these eggs had antibodies, and average was 4 to 5 log 2. And these eggs, of course, they came from non-vaccinated hens, no antibodies. And what they did, they inject the F98 strain in these eggs, right? So after that, after 3 days, they harvest the F98 strain from this first generation of eggs, and they inject the supernatant in the second generation of eggs. And then they wait for 3-4 days, they harvest the virus here, and they inject in the third generation. And they did it for till the generation number 80. So you imagine the amount of work. And at the end of the day, what they want to see is that the change in the virus in these two different groups, coming from vaccinated eggs or virus replicating eggs that have antibodies or virus replicating eggs without antibodies. And they measure several things. I would just keep the big conclusions of this paper. But they conclude that the passage of H9N2 virus with or without selective pressure of antibodies showed genetic variation, enhanced replication, and variant antigenicity. So the virus changed. With or without pressure of antibodies, the virus changed, right? However, and this was something very interesting, the selective pressure of the antibody does not seem to play a key role in the antigenic drift in the egg model. So very interesting that in this model, it seems that the changes were not very much significant when they compared the virus in eggs with antibodies and eggs without antibodies. Right, but you can say, well, but this was done in eggs. This is not really a model that we could rely on. And then another group, different group, they published this year, very recently, in 2020, they published something very similar. They used the same vaccine or the same experimental vaccine from a strain F98. They immunized some SPF chickens, and there was a group that was not immunized. So likewise, they infected these chickens by nasal drop or eye drop with this strain. After a few days, they harvest this strain, they prepared the inocula, and they infected the supernatant in the second generation of chickens. Again, they did it in the birds that were previously immunized, and birds, SPF birds, were not previously immunized. So they just measure the difference the virus in these two types of birds. And they did this till the generation 20. So they passed this virus 20 times in birds with antibodies, active antibodies, and they passed this virus in birds, in SPF birds, without antibodies. And again, it was measured in the several criteria, but what they found out was, interestingly, it seems that the selection pressure of the vaccine antibodies plays a role in regulating and controlling the gene mutation of H9N2 avian influenza virus. This was very interesting because you would expect that the virus would deviate, would mutate, would even become more pathogenic, but no, it was the opposite. And they measured this in eggs, and the egg lethal dose in the virus from the vaccinated disappeared. This virus was not able to kill eggs anymore. So these authors, they mentioned that it seems that the antibodies play a role in regulating and controlling the gene mutation, right? And finally, the last one also comes from Korea, and this is more comprehensive study, and it investigates the evolutionary dynamics of H9N2 viruses circulating in Korea from 1996 to 2012. You remember that the vaccination was introduced in 2007, right? So they introduced the vaccination here, and the immediately effect was the clade where the vaccine came from disappeared immediately. This was very interesting that one of the clades disappeared, so this was a very positive result from this vaccination. But they went further and they made several analyses, and what they conclude is that the analysis of viral population dynamics revealed an increase in the genetic diversity between 2003 and 2007. Remember when the vaccination was introduced, and then a sudden decrease in 2007. You can see the decrease here. An implementation of the H9N2 vaccination program in Korea has dramatically reduced the diversity of H9N2 virus. Okay. Another point that these authors also mention that is the periodic update of the vaccine seed strain should be considered to maintain the control of H9N2 low pathogenic avian influenza outbreak. So again, three different ways, and it's three different publications show that the vaccination would not increase the mutation rate. However, for the sake of scientific fairness, this is not a consensus, right? There are papers, there are authors that do believe that vaccination might increase the mutation rate. I tried to find this with H9N2. I could not, but I was able to find some publications with was H6N2 influenza. They evaluated the evolutionary consequences of a decade of vaccination in South Africa, and they really think that the long-term vaccination may contribute to some phenotypic change of the virus, right? And another publication, this is an older one in 2011, but the group of Giovanni Cattoli, they compared the H5N1 virus from countries that were not vaccinating and countries that were vaccinating against H5N1. And it seems in that publication, the virus in countries that were vaccinating had a greater differentiation or greater mutation as compared to the virus circulating in countries that were not vaccinating, right? So just for the sake of scientific fairness, this is not a consensus among the scientific community, right? Now, I'm coming to the end of my presentation, and I would like to have some conclusions or some food for thoughts. And in countries where H9N2 virus is enzootic, some measures should be considered. As I mentioned before, eradication is not feasible with the tools that we have today. So we really should consider some points. And as I mentioned before, strengthen up the biosecurity procedures. Probably this is the most important of them all, but also to improve protection against other respiratory pathogens like IBV and NDV, ILT, and G. E. coli, or even immunosuppressive agents. Gamboro, CIAV, or mycotoxin. Third point, avoid excessive respiratory vaccine reactions and improvement of environmental conditions. This would help to decrease or to minimize, to mitigate the impact of the H9N2. We have to keep a close monitoring of the virus to see mutations, if the vaccines are necessary to be changed. That was mentioned by that publication in Korea. And we need to keep monitoring because we need to see whether this virus is spreading to different regions in a given country. So monitoring is rather important. And finally, vaccinate the flocks against H9N2 with high quality vaccines. And at this point, we need to say, if approved by local authorities, right? So this is what I had prepared for you guys. And these are all the papers, all the articles that I mentioned during this presentation. I will send them all to our Indian colleagues, and you can contact them if you are interested to go deeper in any of these issues. Okay? And with that, I finish my presentation. I thank you very much, and let me stop sharing here to be able to see you. Thank you very much, guys.
Dr. Marcelo, thank you very much. It was very clear about the main strategies of H9N2 and the solutions. So I would like to welcome all of our speakers to the stage in order to start the Q&A. I'm very happy that we have plenty of questions, but please, you can continue asking your questions. And meanwhile, I will be ready to read all of them. So I will start from the first question, and I believe everybody can hear all the speakers. So what is the age sensitivity or resistance for H9N2 in broilers?
Okay, so I will take this one. So up to my knowledge, there is no reported age resistance to H9N2. So basically, they are susceptible all along their life.
Thank you, Dr. Christoph. All right, clear. The second question, how to counter the severe egg production losses in layers affected with low pathogenic avian influenza?
So the question is very broad. I mean, as far as I understand it, is how to prevent the egg drops in layers due to H9N2. So then it's driving us to the overall prevention strategy that has to be applied to prevent H9N2. And it has been developed especially by Dr. Marcelo in his last talk. And it's a full set of overall preventive measures, which are starting with biosecurity. And we know that H9N2 as a virus is not resident in the farm, is not that resistant, contrary to Gumboro virus or chicken anemia virus. So it comes from outside. So it comes from our mistakes, if I may, in terms of insufficient biosecurity. So we have to do our best in strengthening biosecurity through traffic control, through movement control, people control, as well as wild birds control, among other measures, to make sure that this virus is not entering into our farms. And right after that, I would say it's about mitigating the possible cofactors of H9N2. And they start by applying a very sound prevention program against Newcastle disease, against infectious bronchitis, against infectious laryngotracheitis, mycoplasma, E. coli, you name it. But just to reduce the overall stress to the respiratory system that can occur when H9N2 unfortunately pops up in the overall environment. And obviously, for those countries where H9N2 vaccination is authorized, then we can put H9N2 vaccine on top of that.
Thank you very much, Dr. Christoph. Very clear. Another question is coming about before serological tests, can we draw a preliminary conclusion about H9N2 virus from symptoms and post-mortem findings in field conditions? What are the main symptoms or the signs?
I would say it's pretty difficult. It's pretty difficult because as you saw, even though I had no time to display very extensive amount of pictures, but there is no real typical or pathognomonic signs of H9N2. It looks like very much an upper respiratory tract infection or a condition that's inducing egg drops. So I would say it's pretty difficult to draw a definite diagnosis purely based on clinical manifestations and post-mortem findings. Perhaps only one single example is the observation of these fibrinous plugs in the bronchial bifurcation that is quite strongly suggesting a co-infection of H9N2 together with bronchitis virus. So I would say perhaps this sign is pretty strongly suggestive of H9N2 with IB.
Very clear. I fully agree with you, Dr. Christoph. I have just received the latest question, maybe more relevant to that last question. So there are few outbreaks of H9N2, particularly in vaccinated flocks. Shows only ovidic wall edema with production drop. No respiratory signs, whether such outbreaks are due to drift of variants or antigenic differences in vaccine virus and the field virus.
Okay, so it includes several aspects. First of all, we have to check what was the vaccination program, which vaccine was used, how many applications were done, how the vaccination crew was trained in order to apply the vaccine properly. I mean, just simply applying one full dose per bird. So there are many aspects to consider. And secondly, obviously, is to make sure about the diagnosis. So whenever there has been any clear confirmation of H9N2. Lastly, and I think it was also very clear from the presentation of Dr. Marcelo, we can still get some H9N2 detection even though we do vaccinate. Vaccination will not lead to eradication of H9N2, so it can still happen, can still be there. However, the aim of applying biosecurity, prevention of cofactors, and vaccination, if it's allowed, is to reduce the consequences of H9N2. It's definitely not to eradicate it.
Understand. Thank you very much. One question came. Can you specify the Indian clade?
I guess that's for Gwen, probably,
Yeah.
to confirm that.
Yeah.
She mentioned that, but just to
Yeah,
confirm.
Western G1.
Western G1.
Yeah.
And another related questions maybe, do crows play a role in H9N2 virus as an India avian infection virus is seen in crows? So it may be confusing with nest wild virus or not?
Yeah, so far, to my knowledge, mortality in crows has only been related to H5, HPI viruses, not only in India, but in other countries. It doesn't mean that crows cannot be infected with H9N2, but they would probably not die from it because it's a low-path virus. So crows may play a role in transporting H5, but it may not be the main species. Ducks are really the natural reservoir for avian influenza viruses in general.
Thank you, Dr. Gwen. Another question was asked about the reported case from India about H9N2 and the labs.
Yeah, so I think I saw the question whether Bhopal has been able to report
Yes.
some cases, and indeed, I mean, they published a few papers but it was already about 10 years ago. I guess they need new samples to be able to publish, but we had Dr. Nagarajan and Dr. Tosh among the the authors, and there were indeed publications describing some H9N2 infections and phylogeny of these viruses, but these need to be updated.
All right. Thank you very much. A question to Dr. Marcelo. Can we diagnose the primary cause in the field if there is ND and H9 at the same time with real-time PCR?
Which virus came first, ND or H9? If you have RT-PCR positive for both of them, I think it's very difficult to make any assumption that H9 or ND came first. It's just by chance. The two possibilities could happen.
Makes sense. Of course, a relevant question for all of you may be that what should be the vaccination program of H9N2 in broilers, where birds are harvested in 33 days, more or less?
This is a very, very interesting question. It's a very controversial topic, actually, because usually if these broilers are raised in exotic areas, the breeders are heavily vaccinated or even vaccinated and infected. So the level of maternal-derived antibodies will be very high. So if I vaccinate these birds in the hatcheries, there will be interference. So I have to delay. If I want to optimize the vaccine take, I have to delay and to do it in the farm. However, if I do it, I will take the risk of my vaccination crews being the agent of bringing the virus to that population. So it's very difficult question to answer, and it's not a consensus. Why? Because the killed vaccine will list humoral antibodies and will never stop the infection. So even if you have vaccination, if you have good vaccine take, still you are not going to be able to stop the infection and the consequences of that infection in broilers. This is a matter, is a good debate, I would say, but there is no consensus.
Then another question to you, Dr. Marcelo, and of course to our other speakers. Which vaccine will be more effective, H9 alone or combined with Newcastle disease?
To H9, I assume there is no difference. Usually, and then I have to speak for the vaccines that I know, that range from Ceva. The amount of virus in the H9 monovalent vaccine is absolutely the same amount of virus in the combined and the H9 vaccine. So, and we have vaccinated, there is no significant difference. So talking about protection against H9, you can use both. There is no difference.
Thank you very much. What is the recommended age for vaccinating H9 killed combined? And what should be the effective gap for a booster dose?
Well, I'm assuming that this question is coming to me. Usually for H9, and I'm going to speak about countries that we are using these vaccines here in Asia. For H9, the recommended age for long-living birds, the layers and breeders, is to have 2 shots, at least. In some cases, our producers like to go for the 3rd shot, but usually the first shot is around 6 weeks of age, given or taken, and the second shot for 2 to 4 weeks before the onset of production. What we observe in the field is that some producers are seeing a very early infection. So they take the opportunity of the big trimming and they inject half a dose at around 7 days, 8 days. And then they go for 6 to 8 weeks. Second shot, and then 2-3 weeks before the onset of laying, another shot. So it varies, but basically is that. For H5, and the vaccine that we commercialize in Asia is a vector HPT-based AI vaccine. And this vaccine has to be given in the hatcheries. Okay, but when you go for killed vaccines, usually they follow similar vaccination recommendations, as I mentioned, for the H9. Okay, but we don't commercialize this type of vaccines here in Asia.
Thank you very much.
If I may just add a comment, sorry, on Marcelo's point. What is described here is what we are recommending at Ceva, because we know what are our vaccines and what's the formulation of our vaccines. But for the audience, it's very important that you ask this question to any of your supplier, because actually the quality may vary quite a lot among vaccine suppliers in terms of antigenic concentration, strain, antigenic match with the field isolate, adjuvant quality. So there are many, I would say, composition elements that will bring you towards a high quality vaccine or to a medium to low quality vaccine. And obviously, with a rather low quality vaccine, you will need much more shots in order to get a similar effect. So bear this in mind. There is a quite huge variability among vaccine producers.
Thank you very much. It's very valid comments also from Dr. Christophe. I believe we answered some of the other questions about vaccination program, because it was coming, what is the recommended vaccination program from 1 day till 60 weeks in breeders and the broiler breeders. Should there be any revaccination in the mid-lay? Maybe we can also answer this question as well.
Yes, this is been a very common question in the countries that are affected by H9, and some producers follow the seroconversion, the antibody response of the vaccine, and if they see any reduction, what they consider their baseline, they like to go for a revaccination mid-lay. What we have seen is that when you do it, you might have the risk of causing some slight drop in egg production, not because of the vaccine, but because of the handling of the birds mid-lay. So you have to make this decision with the veterinary of your company, because you might have this kind of drop in egg production because of the handling.
Thank you, Dr. Marcelo. Another question comes about the onset of immunity and duration of immunity of these inactivated killed vaccines.
Onset of immunity, this is for very broad, but usually for killed vaccines, you're going to start to have detectable antibodies 10 days, 2 weeks, and you're going to have 3 to 4 weeks, the maximum level of antibodies after the vaccination. If I could say that this seroconversion or antibody response is what you call the onset of protection. And duration, it's a little bit more difficult to establish, because we can establish the duration of the antibodies, right? But it's very difficult to establish that if that level of antibodies will still be protective. As I mentioned before, and this is important to reinforce, that these vaccines will not prevent the replication of the virus. So if you consider protection as replication of the virus, so it's difficult. So we have to define protection before measuring it. I hope I could explain my thoughts.
I believe so. Another interesting question: why we couldn't produce a live vaccines for avian influenza, especially for the protection
very good
chickens
question.
we are observing?
Well,
Okay. Actually, in public health, they use some live vaccines. These are applied to the nasal route, but in the animal reign, it wouldn't be recommended to use these. First, for HPI viruses, this is absolutely not recommended for sure. And for low path, there's always a risk for the virus to then reassort with other viruses. Avian influenza viruses are so dynamic, so we wouldn't want to create this kind of mess, which is already naturally occurring. So it is not recommended at all to produce the low path viruses. I mean, low path viruses, live viruses.
Attenuated vaccines.
Yes, that's what I meant.
Then, after touching this point, so can you also give another type of vaccines for avian influenza? So inactivated vaccines, and also you have vector-immune AI, right? The vector vaccines.
Yes, you can combine the use of vector-immune AI that you apply at 1 day of age with inactivated vaccine. It is always good to combine different technologies because it provides complementary immunity. The vector-immune AI is very much a virus that would elicit cellular immunity, also some humoral immunity, but minor. And inactivated vaccines are very good at eliciting humoral immunity. So if you start with one vaccine and you boost with inactivated vaccines, then you would boost the humoral immunity with inactivated vaccine, which could be a very high interest, especially for breeders, in order to get maternally derived antibodies in the progeny.
Yes,
Just to be clear for every.
Yes. Go ahead,
Sorry, Marcel. Yeah, just to be clear for everybody, vector-immune AI is against H5 virus, okay? And there is no cross protection between H5 and H9.
That would be my comment, Christophe. Thank you.
Thank you very much for this clarification. It's very important. Another question is, does H9N2 alone producing hemorrhagic tracheitis, so damage on the respiratory tract?
So I guess that question is for me. I would say yes, it could happen. Since in biology we usually never say never. Indeed, it's not very frequent. However, in our scientific campus, we tested several isolates as a potential, challenge strain option, and indeed, among candidates that we took from an area covering Morocco up to China, we could find some isolates that were more pathogenic by themselves compared to others. So if we completely exclude the environmental conditions, the ammonia level, the stocking density, litter quality whatsoever, and the secondary microorganism, it may happen, yes, indeed, according to the prevailing strain, that H9N2 alone is inducing some hemorrhagic trachea, yes.
Thank you very much, Dr. Christophe. There is an interesting question that there are many outbreaks facing in the fields by our customers, and the question is how to convince Indian government to allow the use of bird flu vaccines. Dr. Milins, maybe you would like to answer?
Yes. Yeah, he's frozen.
I think he's frozen.
Maybe we can take another question until he's back.
Exactly. So another question is coming about day one broiler vaccination will be useful for protecting the birds up to 35 days of age. Is it enough or not?
Yes, this is what I answered before. If this broiler is in an endemic area, most likely the breeders will be heavily vaccinated or even infected, and they will have high level of antibodies, the passive immunity, and this will have an interference with this vaccine take. So if that is the case, most likely it won't protect the birds.
That's the main paradox, I would say, of H9 vaccination in broilers, because there is this maternal immunity, which is very helpful. So we like to get as many breeder immunization and titers in breeder stock as possible to be transferred to the progeny. On the other hand, it is very significantly preventing the proper immunity enhancement by a killed vaccine when it's given at hatch. So that's the issue of successfully vaccinating broilers. Yes, indeed.
Thank you very much. Another interesting question that what should be, Dr. Milint, are you with us?
Yes, I'm back. I'm sorry.
Okay, so maybe I can remind the question again to you that how to convince Indian government to allow the use of bird flu vaccines?
This is very important question, and I fully understand the gut feeling behind this question. But let me explain to the audience that please don't be under impression that Indian government is absolutely insensitive to this issue. I have been discussing closely with our policymakers and bureaucrats, and more particularly with our Animal Husbandry Commissioner. And there is a very sincere thought process, brainstorming that is going on. However, they are taking a very scientific approach to address this problem. You know, it's not that easy that you just import the vaccine and start vaccinating those birds. In spite of representing Ceva, which is a vaccine company, I must tell you that there has to be a scientific way. We should know what kind of a strain is there, what kind of clades are there, what is the antigenicity, what is the molecular makeup of the strains prevailing in our country. Unless and until we don't do that, it will be really very risky to bring in some vaccine. And trust me, Indian laboratories, particularly Bhopal lab, Nishad, they are on the top of the whole study that is going around, and they will definitely give the right feedback to the government, and soon government will decide the policy. How to go for vaccination, if at all, what is the current situation about the strains prevailing, and what kind of vaccine will be suitable for our country. It is definitely going to happen. Our government is not at all insensitive to this. Thank you.
Thank you very much, Dr. Milint. We can understand the needs and also the intention as well. So we hope the best solution for India soon. Another question: if a layer flock already exposed to low pathogenic avian influenza, and the flock have protective titer 5,000 level, then what consequences should be happened? What can we do? What is your recommendation about this situation? I can repeat if you want, or
No, to whom?
I'm not sure I understood the question actually.
okay, let me explain again. There are too many questions similar like that. So there is a layer flock exposed to low pathogenic, and the mean titer is already high. So what can be the consequences after that, and what is our recommendation on that?
Well, if it's
Yes.
already in production, I mean, it depends whether there is an impact in production of this H9N2 infection, or if it has been found, let's say, by chance, something like that. Now, if we are facing an outbreak with drop in egg production, there is no magic tool that can be applied except giving some, supportive medication, vitamins, things like this, in order to help these birds recovering quickly.
I understood the question a different way. If the flock had been exposed to H9N2 and maybe faced drop in egg production, should this flock be vaccinated using H5N2 vaccine afterwards, knowing that it has a level that they consider good level of antibodies? This is the way I consider. And I don't think that the vaccine afterwards would help anything. If the flock will be exposed you pay the consequence of this exposure, but then it will be kind of protected because of this exposure.
Very much. So we still have some questions, but lastly, one important question is, well, I will combine two questions together. So what should be prevented first for H9N2 or the respiratory cofactors? That's the first question. And the other one is, can we face more than 2 respiratory pathogens together? So, Dr. Christoph, would you like to take this question?
Okay. So, all right. So for the first question, whether we should target first H9N2 or the cofactors, I think that it's probably easier to start by the cofactors because they are already well-known strategies and well-accepted and authorized strategies to do so, which start by applying strong biosecurity management enhancement, as well as prevention of immunosuppressive diseases, and we can think about Gumboro first, as well as the main other respiratory diseases like Newcastle disease, infectious bronchitis, laryngotracheitis, among others. So I would say that if all of these issues are very well controlled and I would say out of the farm, any H9N2 infection will induce much less consequences because it's going to be alone compared to a situation when it's combined with the other ones. So that's the first point. And the second question is whether we may face more than 2 co-infections. Yes, unfortunately. We are currently running a kind of study in Northern Africa. And this is one of the first conclusions of this survey. When we use, let's say, swabs from acutely infected birds, that until now we could find up to 4 concurrent viruses or bacteria, or virus and bacteria, playing a role together in this acute upper respiratory tract infection. So yes, indeed, we can get more than 2 culprits into this respiratory syndrome.
Thank you very much, Dr. Christoph. Well, of course, we have some other questions, but I would like to end up here. We will be reaching you our questions, the answers afterward. So if we need to conclude, so I would like to thank all of your patients for the last 2 hours we have been together. We have 3 presentations. The first was about the worldwide perspective on H9 viruses by Dr. Guvenel. So we understood the endemic and the situation of H9N2, not only in India, not only in Asia, but worldwide for many years. And then the second, Dr. Christoph has talked about the importance of H9N2 in 4 pillars, in terms of genome, in terms of coming complication with other things for zoonoses as well. And then at the end, Dr. Marcelo has talked about the main strategies and what we are doing about it. So I believe you have liked. So with the light of this information, I would like to invite you to the survey once this webinar is ended. So you will be guiding us for our future webinars to improve and to touch much more relevant topics. So I would like to give the floor to Dr. Anand once again to make his final speech. Thank you very much. It was a pleasure for being you and looking forward to have another ones in person. Dr. Anand?
Thank you, Dr. Mustafa. First of all, let me thank you on behalf of Ceva for taking time out of your very busy schedule in the afternoon and joining on this webinar. I hope that you found this very interesting and useful. There may be many questions in your mind which we were not able to answer today. Please reach out to us either through our sales team, technical team, or the service engineers, or directly to us, and we'll try our best to answer these questions. We will continue to have a series of these webinars in future, and if there is anybody who missed this webinar because they were not able to join because of any network issue or anything else, please request them to reach out to us also, because we will try to share the recorded version on request. So with that, again, thank you very much, and thank you for all the speakers and the participants for your total participation and engagement during this webinar. Thank you very much, and have a good day.